The liver is the only organ in the human body that can regenerate. But why? Why not the heart or the brain? Those seem way more important. To understand why the liver can regenerate but other organs can't, we need to understand how regeneration works. For regeneration to occur, you need to have two things. Firstly, you have to create new cells. New cells can be produced either by a cell dividing itself and creating an identical copy, or by a stem cell which can create a new mature cell. Stem cells work by dividing themselves and creating a specialized daughter cell. So now, after having created a bunch of cells, you need to be able to give the cells specific instructions that tell them where to go and how to act. A bunch of cells simply won't be able to generate a new arm without instructions guiding them. So now we know that regeneration requires two things: creating new cells and instructions to tell the cells where to go. Let's look at the first point of the checklist: creating new cells. I mentioned earlier that new cells can be created either by dividing themselves or by stem cells creating new cells. The process when cells divide themselves is called mitosis. What happens here is that the cell copies its DNA and then splits itself in half, creating two copies of the cell with the same DNA. Now, this process works quite well at creating new cells and is used by the body all through childhood. But when the body reaches adulthood and stops growing, the cells enter a resting phase and stop dividing themselves. Another way the body creates new mature cells is through stem cells. Stem cells also use mitosis to divide, producing two daughter cells. One of these daughter cells remains a stem cell, allowing the body to keep a supply of stem cells for future repairs. The other daughter cell becomes more specialized and develops into a mature cell that performs a specific function. Adult stem cells cannot become any cell. They can only become cells that they are specialized in. Okay, so now we have two different ways to create cells. We only need instructions and we're good to go, right? Well, yes, but the instructions are the hardest part. Growing cells is easy, but telling them where to go and organizing them is incredibly hard. Normally, cells in the embryo unlock specific instructions from their DNA. These instructions are very detailed and rigorous. They essentially tell the cell exactly where to be, what to be, and how to act. But after birth, the instructions in the DNA get locked and are inactive, which is precisely why humans cannot regenerate. Our bodies can create millions of cells, and we can even create cells in a lab, but the hard part is getting them organized. But why then can the liver regenerate? And why can't other organs? Firstly, we need to understand what the liver does. The liver is used to detox the blood. This means that waste, toxins, and anything harmful goes through the liver. This means that the liver is constantly under stress and needs to be able to repair itself. So, liver cells called hepatocytes have this very unique feature where they can exit out of the resting phase and return to the cell cycle. This means that liver cells can undergo mitosis and duplicate, and they're incredibly good at doing so. Liver cells are usually described as very proliferative, meaning they can increase in numbers incredibly well. But how do the cells know where to go? Well, using chemical signals from nearby cells and blood vessels, the liver cells can organize themselves. But why can't the heart or other organs regenerate? Firstly, a lot of organs simply cannot create new cells or have an incredibly hard time doing so. Usually, cells that are specialized cannot be replaced. For example, heart cells and brain cells are incredibly specialized cells. Everything from their structure, shape, position, and form is incredibly important for their function. In those cases, replacing them is not viable. Brain cells, for example, are incredibly specialized. Each neuron has a very specific job and is connected to a bunch of other neurons. This means replacing a brain cell requires a complete new rebuild of the surrounding brain cells. Okay, but what about other organs like intestines or limbs and fingers? Cells in the finger aren't as specialized like cells in the brain or the heart, but the structure of a finger is very complex. You need bones, ligaments, and skin all in the right places. If you try to regenerate a finger, you would most likely end up with a cancer-like tumor instead of a proper finger. A good analogy that explains how the liver can regenerate is to imagine you have a balloon filled with tiny beads inside, each bead representing a cell. Now imagine removing half the beads and the rest is filled with air. You could probably still fill the balloon back up with beads while maintaining the same shape. The same goes for the liver. The liver is filled with blood vessels, matrices, and surrounding cells that all guide the new cells on where to be. In conclusion, the liver can regenerate because of two reasons. Reason number one is that liver cells are incredibly good at dividing and creating more of themselves. Reason number two is that the liver's complex network of blood vessels and surrounding tissue stays intact when cells are lost, acting as a scaffold that guides new cells back into place.